piece of paper and have a pencil. You draw the picture in your mind. What's the structure of the bacteria have? We go from there. It's like an outline. Then you will know this chapter very well. So we want to talk about these things a little bit in detail and one by one. Okay. Bacterial cells envelope, they call it plasma membrane, cell wall outside layer. So we talk about these one by one. Something you know, but something we want to be emphasized. Okay, first of all, cell membrane structure. This is everybody knows. Fluid mosaic model. By the way, fluid mosaic model is 1972 by two American scientists. Singer, one of them was Singer, another person I kind of forgot. Both of these two scientists is a professor in California. One of them is in University of California, San Diego. Another guy is in medical institution of California, somehow in San Francisco. In 1972, they pointed this fluid mosaic model, will explain the cell membrane system. So what it looks like, said very simple, which is a protein and phospholipids, a double layer, we will say a double layer, integrated in a mobile status. So they actually is integrated with each other in a mobile moving status. It's not like static state there. So what it looks like, we draw on the picture already, so I'm going to draw here. Okay. It's simple, but something we have to mention. Okay, you have these phospholipids right here. We know these things. And then you have a protein right there. That's called fluid mosaic model. It's always like this, simple. So we want to talk about these details, and you can explain, you can understand more well. First of all, regarding the protein, there is basically two types of the protein there in the fluid mosaic model. One of them is in the middle across the phosphate double layer. This one, like a channel, we call integral protein. Okay, how many are there? Around 70 to 80%. But how about the others? Majority of the others is right here on the side. It's right here. Somehow here. It's like attached to the integral protein. This is called ferroprotein. This is around 20 to 30%. Peripheral protein. But why they have to be there? And I tell you it is important. Because these are the places later on will happen is ATP hydrolyzed become ADP and the phosphates. So this is an ATP binding domain. So you know that most of the time when you hear the terminology domain, basically it's a protein. So that's why this side chain, this peripheral protein is actually important, although it is only about 20 to 30%. So that's the first thing is a protein. What else we have? You think fluid mosaic model is so simple, but it's not really. Sometimes you will see something like this, like a chain showed up. As could be a sugar, oligosaccharides. And sometimes you also will see something is Y shape. What's a Y shape looks like? Also, there's a couple of circles there. There could be a combination of sugar and fatty acids, so we call it glycolipids. Sometimes the integral protein is not that thick, not that heavy. It will be very thin, just a pass it. And in the middle is alpha helice. You're going to see that. And also you're going to see something right in the middle like this. This is hypernoid. Why it is important? This is, we call it a modified cholesterol, because for some of the environmental bacteria, this is for petroleum generation. I want to mention this here is important. West Virginia area is mining, little bit of stealing natural gas, petroleum. I would be assuming in our ground, there are some of the environmental bacteria could be contributed to the natural petroleum gas. Could be somehow, although it's not a major contribution. But we would say some of the natural microorganisms have those petroleum generation. It's interesting. So that's something. Okay. Now next we want to mention this lipids. This sounds very easy. Everybody knows it. Why are you gonna spend time to talk? Because some of you will be always confusing about the head and the tail. Okay, this is the head. We call it a hydrophilic. It's water-like. And this is the tail. Hydrophobic. Is water dislike. But sometimes you messed it up. When I ask them to label, they always label something, people label opposite. Now how you understand it? The reason is you labeled it wrong because you only remember it. You don't know the story behind it, why it is. But that's related to the chemical structure. So we need to explain to you. Why the head is hydrophilic, why the tail is hydrophobic? What's the tail looks like? NH3, CH2, CH2. What is this one? What is CH3, CH2, OH looks like? Alcohol, is that right? Yeah. I'm going to replace the alcohol with amino group. What's the name of this guy? Ethanoamine. Can alcohol dissolve in the water? Yes, of course. Otherwise, we want to make 95% alcohol. So, this is dissolved in the water. Did you learn amino acid structure? When you see the amine group have a plus there, what this means? It's a polar, is that right? It's a polar structure. So water-like. Okay, therefore, hydrophilic, water-like. Because there is an ethanoamine on the top. What's going to be here? Now, obviously, we saw it's phosphates. Double layer, we need a phosphate. No question about it. We need these phosphates. Okay? This is simple phosphates. So what's the middle? Need a C, H2, CH, CH2, and go here, CH, go O, CH2, 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 CH2. What is this guy called? Glycerol. Is that right? That's glycerol. Then, what is this guy here? That's CH2 called B16. Fatty acids. What is a fatty acid? Like an oil. When you buy a bowl of soup somewhere, did you see that floated on the surface? Is that dissolved in the water? No. Because the bottom is fatty acids. And also the glycerol connected to that. So this is dislike water. If it's dislike water, you didn't see any charge there, it is nonpolar. Now by the way, if it's 16, what it is? Aromatic acids, is that right? If CH2 goes there, is 18, what it is? Stearic acids. That's people tells you don't eat too much fat because stearic acids is a saturated fatty acids, not easy to be dissolved in the liver area. Finally, will become cholesterol. The cholesterol will be attacking your heart. You have a blood attack, a blood clotting, generate all kinds of side effects for the health. Is that right? So when you draw the picture here, what tells you, you will understand why this is going to be looks like that. Otherwise, you're only going to remember, because on the top of the head is ethanolamine. It is dissolved in the water, and the bottom is a long-chain fatty acids. It's like oil on the top of the water floated. It's this like water is a non-polar system. That's why hydrophobic. Okay, so that's something. It's very interesting. We talk about fluid mosaic model. So remember this, and in the exam, we will ask you to label the top and the bottom, the some structure of the cell wall. We won't ask you to do this. Okay, don't worry. This is organic chemistry, but we explain to you. Okay, so that's somehow behind the story. Okay, Thursday we'll talk about the cell wall structure. That will be the main part.